previous lecture, we discussed about the process of sperm formation, gametogenesis, that is spermatogenesis. We discussed like what are the stages of gametogenesis. Spermatogonia, it undergoes multiplication, and first phase is multiplication phase, next phase is growth phase, third one is maturation phase, the last one is differentiation phase, right? So the process of spermatogenesis we discussed in the previous lecture. In this lecture, we'll talk about the structure of the sperm and hormonal regulation of gametogenesis, spermatogenesis. We'll see. First, we'll look into the structure of the sperm, right? So in our today's lecture, so we'll talk about the structure of sperm. So I drawed it. There we can see the structure of the sperm, diagrammatic representation. Now, human sperm was discovered by Leeuwenhoek, right? Who discovered human sperm? Sperm was discovered by Leeuwenhoek. What is sperm? Sperm is a male gamete. It is a male gamete. And who first saw human sperm? Human sperm was first observed by, it was first observed by Leeuwenhoek. Right? Now, if you see the shape of the sperm, what is the shape? Is it like a tadpole shaped structure? It is a tadpole shaped structure. We can tell it's a dart like tail structure. It is a tadpole shaped structure. Or we can also call dart like, right? Formed by a process called spermatogenesis. Now, what is the length of the sperm? What is the breadth of the sperm? Each sperm measures about 60 micrometers in length and 3.5 micrometers in breadth. Each sperm, how much it measures? 60 micrometers in length, and it measures 3.5 micrometers in breadth, right? So this is the dimension of the sperm. And where is sperm formed? Sperm is formed inside the seminiferous tubule. Where is the seminiferous tubule? Inside the testicular lobules. Where are testicular lobules? Inside the male gonad called testis, right? So sperm is produced by a process of spermatogenesis inside the seminiferous tubule. Sperm, it is a male gamete. It is an animal cell. Animal cells will not be covered by cell wall. So they are just covered by plasma membrane all around. So no cell wall. They don't have cell wall. And it is wrapped by plasma membrane only. It is wrapped by plasma membrane because it is an animal cell. Cell wall will not be there, right? Now, what are the portions of a sperm? If we look here, so 1, 2, 3 and 4. So 4 prominent regions are there. This much portion which is anterior, so which is broad, which is flat, is called as head. Below the head, there is a narrow, short region called neck. Below the neck, there is another region called middle piece, in which we can see mitochondria there. And then the longest part of the sperm is the tail. So what are the 4 regions? So it has 4 regions. It has 4 regions. One is the anterior head. Below that, narrow neck. Below that is a middle piece. And the last one is the tail, right? Now, it is a dart-like structure, it's a tadpole-like structure, it is uniflagellated. One flagella is there and it's a motile gamete. So, it is having one flagella. Because of this flagella, it can swim, it can move. So, it is a motile male gamete. Now, when we have to talk about each and every part, the first part is the head. So, we told it is anterior, it is broad, it is flat, means it's the flattened part, and it shows a cap-like structure. This cap-like structure, which is above anterior, is called as acrosome. You can see inside the acrosome, it's filled with lytic enzymes, and this acrosome is derived from Golgi. Golgi body will secrete this acrosome. So anterior, it has a cap-like structure which is called as acrosome. Below the acrosome, it has haploid nucleus, male nucleus it has, right? So what can we write about the head? Head is anterior, right? And it is flattened. It is blunt. It is not pointed, right? It is blunt. It is blunt. It is broad also. Region, right? Now, it shows a cap-like structure. It shows a cap-like structure. What is it cap-like structure called? I told it is acrosome. It is acrosome. This acrosome is filled with lytic enzymes. This acrosome is derived from Golgi. Acrosome, from where is it derived? It is derived from Golgi. And it is filled with, what is it filled with? It is filled with lytic enzymes. Acrosome is filled with lytic enzymes. These lytic enzymes can dissolve the membranes which are around the egg, so that the sperm can easily penetrate inside and go and fertilize the egg, right? So, where are all the enzymes present? In the acrosome, the enzymes are present. So, what are the names of the enzyme? We know that egg is covered by hyaluronic acid. Now, to dissolve the hyaluronic acid, acrosome should have hyaluronidase enzyme. Then, corona radiata, one layer is there in the egg. To dissolve that enzyme, it should have corona penetrating enzyme. Zona pellucida, one more layer is there in the egg. So, to dissolve that layer, it requires zona lysine. So, come on, 3 enzymes I have quoted. So, which are required for dissolving the membranes around the egg, so that the sperm can easily penetrate and fertilize with the egg. Where are all the 3 enzymes located? In this cap-like structure called acrosome. 2/3 of the head region is occupied by acrosome itself. Okay. So, I am writing here 2/3 of the head region is occupied by acrosome itself. It contains lytic enzymes. What are the lytic enzymes? So, we have to list 1, 2 and 3. The first enzyme is called as hyaluronidase. Egg is surrounded by hyaluronic acid. To dissolve that hyaluronic acid, it requires hyaluronidase enzyme. Hyaluronidase enzyme is also called sperm lysine. What's the other name? It's called sperm lysine. The second one is corona dissolving enzyme. Corona penetrating enzyme. Again, I am telling, like egg is having a layer called corona radiata. So, through that, if the sperm has to penetrate, it needs a corona penetrating enzyme. And the third enzyme, egg also has zona pellucida. To dissolve that zona pellucida, it requires zona lysine. So, what is the third enzyme? It is called zona lysine. It is also called acrosin. The zona lysine is also called acrosin. Amma, where are all these enzymes present? They are present in the acrosome. So, they will help in penetration of the sperm into the egg, so that it results in fertilization. So, that is about the acrosome, and below acrosome is the head. Below acrosome, it has a haploid nucleus. It has haploid male nucleus. It has haploid male nucleus. So, this is about the head region. Head, it is anterior located. Head is flattened. It is not pointed. It is blunt. So, make the anterior region blunt. It is not pointed. It is blunt and it is a broad region. It shows a cap-like structure called acrosome. The acrosome is made by the Golgi. Acrosome is filled with these 3 lytic enzymes. Below that is nucleus. Take a screenshot, then we will talk about the remaining parts. Right. Now, when we have to talk about the second region, what is the second region? It is a neck. It is very narrow, and see where is the neck located? Between the head and the middle piece. And what can you see in the neck? There are 2 dots. What are they labeled as? Centrioles. A pair of centrioles are there in the neck region. So, this is a neck region. Neck region has a pair of centrioles. So, the second part is called as neck. It is a narrow, short region. It's a narrow, short region which is between, neck is between head and the middle piece. Between head and middle pieces, isn't it? Right? It is between the head and the middle piece. Now, the neck, what is it filled with? Sorry. What is neck filled with? Neck is filled with a pair of centrioles. Neck has one proximal centriole and one distal centriole. It has a proximal centriole and it has a distal centriole. Proximal centriole, it is free, whereas distal centriole, it joins to the axial filament which runs through the middle piece and the tail. So proximal centriole, we can tell it is free, whereas distal centriole extends. It extends into axial filament. The distal centriole is extending into a filament which is there in the middle piece and in the tail region. That's it about the neck region. Head, it has some functions, whereas neck, we are telling that it contains a pair of centrioles. It's a narrow region. It is between the head and the middle piece. Proximal centriole is free and the distal centriole will make the axial filament. So, when the sperm is penetrating the egg, do you think that the complete sperm will penetrate? Do you think the complete sperm will penetrate, or only till the head region the sperm can penetrate? The remaining region will be left outside only. So that means only one centriole. So the acrosome, the nucleus and the proximal centriole, only children will go into the egg and they help in fertilization. Whereas the mitochondrial part, the distal centriole, the tail and all will not enter outside. So they will be outside only. They can't enter inside. Okay. So that means which centriole enters into the egg? Only the proximal centriole, which is free, can enter inside the egg, whereas distal centriole will be left behind. Now coming to the third piece. What's the third piece called? It's called middle piece. Now this middle piece, so it is wider, right? And it has centrally located axial filament. It has a central line. What is it called? Axial filament. Now, if you see around the axial filament, so they are spirally arranged mitochondria. What are these called? Spirally arranged mitochondria will be there, which is called as Niebuhr-kern. So, around the axial filament, spirally arranged mitochondria are present. Spirally arranged mitochondria are present. So what is it called? Niebuhr-kern. If they ask you in the NEET questions, what is Niebuhr-kern? It's a spiral arrangement of mitochondria which is present in the middle piece of the sperm. It is called as Niebuhr-kern. And then why do we call the middle piece as the powerhouse? Because here mitochondria is there and mitochondria provides energy for the movement of the sperm. So the mitochondria, it supplies energy. For what? For sperm motility. So mitochondria, it provides energy for the sperm motility. At the end of the middle piece, you can see a ring centriole which is called as annulus. So at the end, we can see at the end, ring centriole is present. What is the ring centriole also called? Annulus is located. Children, this is about the middle piece. The last one is tail. Very easy, right? The male gamete structure is very easy, but the female gamete structure is not that easy. So it contains a head, it contains a narrow neck. The third portion is a middle piece in which the mitochondria are there. They are spirally arranged. That arrangement is called Niebuhr-kern. And at the bottom, it has a ring centriole, which is called annulus. The last portion is called as tail. It's the longest portion and it contains axial filament throughout. Tail is the longest part of the sperm. It is the longest part and it contains axial filament. Right? So the tip of the tail is not surrounded by plasma membrane. So here you can see this blue colored one is coming out. That's the tip of the tail. The tip of the tail is not having cytoplasm also inside. It's not having cytoplasm and plasma membrane around it. It doesn't have cytoplasm also, it doesn't have plasma membrane also. What is the function of the tail? The tail helps in swimming. It gives motility. It helps in motility. So this is about the structure of the sperm. So how easy? There are only 4 parts. The first one is called head, which contains acrosomes, which contains the genetic material, nucleus also, neck region, centrioles, proximal will go inside during fertilization. Distal will make the axial filament. The axial filament will extend in the middle piece and the tail. Middle piece provides energy since it has mitochondria, and the tail helps in swimming. That's it. Now, after coitus, so the liquid which is released is called as semen. Now, that semen, it contains, after coitus, how much amount of semen is released? Per coitus, around 3 to 4 ml of semen is released. Now, in this 3 to 4 ml of semen, how many sperms are there? 200 to 300 million sperms will be there per coitus. The amount of semen which is released, in that semen per coitus, 200 to 300 million sperms will be there. Out of this 200 to 300 million sperms, at least 60% of the sperm should be of normal size and normal shape. They should be of normal size. They should be of normal shape, children. What is normal size? We told it should be 60 micrometers long and 3.5 micrometers breadth. That is the size. What is normal shape? Having head, neck, middle piece and tail, all the parts is called as normal shape. Some sperms, they don't have the tail. Some sperms don't have this much mitochondria. Some sperms don't have the acrosome, like that. Abnormal sperms also will be there. So that's why they're telling in this 200 to 300 million sperms, at least 60% should be of normal size and shape. Again, in this 60%, at least 40% should be vigorously motile. From the 60%, at least 40% should be vigorous in motility. Then only it leads to fertilization. Then only the sperm can swim and it can reach the egg. It can aid in fertilization. So, take a screenshot. Right. So, this is about the structure of the sperm. And then we we'll discuss about the hormonal regulation of spermatogenesis also in this lecture. What is a male hormone? Testosterone. Testosterone is produced by Leydig cells. Where are the Leydig cells present? Which are called interstitial cells also, in between the spaces, right? In between the spaces. Now, let's talk about the hormonal control also. Of what? Hormonal control of spermatogenesis. Hormonal control of spermatogenesis. Now, hypothalamus is there, right? What will hypothalamus do? Hypothalamus will release 2 categories of hormones, releasing hormone and release inhibiting hormone. So, the hypothalamus releases a hormone which is called as GnRH. What is the hormone released by hypothalamus called? GnRH. What does GnRH stands for? We will write down here. GnRH stands for gonadotrophin releasing hormone. Gonadotrophin releasing hormone. Who released it? Hypothalamus has released GnRH. Now, this GnRH comes and acts on anterior pituitary gland. GnRH comes and acts on anterior pituitary gland. Then the anterior pituitary produces 2 gonadotrophins. Hypothalamus is asking the anterior pituitary to release gonadotrophins, right? So, anterior pituitary, then it releases 2 hormones. One hormone is called as follicle stimulating hormone. The other hormone is called as luteinizing hormone. So, we will write the hormones on this side, right? So, what does FSH stands for? FSH stands for follicle stimulating hormone. FSH means follicle stimulating hormone. And what does LH stands for? Luteinizing hormone. Now, they produce LH and FSH. Now, what will these hormones do? follicle stimulating hormone where it will come and act, and luteinizing hormone where it will come and act. Now, luteinizing hormone comes and acts on Leydig cells. Luteinizing hormone comes and acts on Leydig cells. Where are Leydig cells? They are also called interstitial cells present inside the testicular lobes. Now, when the luteinizing hormone comes and acts on the Leydig cells, then the Leydig cell will produce a hormone called testosterone. Then the Leydig cell will produce a hormone called testosterone, which is a male androgen, male sex hormone. Now, this testosterone, what it will do? It has two roles to do. To maintain the male reproductive system, to provide the axillary organs and all, axillary ducts and all. That's the first function, to maintain the male reproductive system. Then the second function of testosterone is to help in spermatogenesis, help in sperm production. So, the testosterone will do two functions. What are the two functions? To maintain male reproductive system. So, it maintains the male reproductive system, that is one function. It helps in spermatogenesis, that's the second function. What's the second function? It helps in sperm production, spermatogenesis. So, the testosterone helps in spermatogenesis. This is the second function. Now, if the testosterone levels are going high, like if I just had my lunch and if I'm not feeling hungry, so then I can't eat it, right? Even if you provide the favorite item what I like, then also I cannot eat it. In the same manner, if the testosterone levels are high, it can stop its own production. It can stop its own production. How it will stop its own production means high levels of testosterone. What is that? High levels of testosterone. They have feedback inhibition. They go and act at this point, hypothalamus. They go and act on hypothalamus, right? And they exert a negative regulation. They can ask the hypothalamus to not to produce GnRH, or testosterone can go and act on anti-pituitary also. Here also negative regulation. It can go and act on anti-pituitary also, and it can stop the production of LH also. So, testosterone if the levels are high, so what is this called? Feedback inhibition. Feedback inhibition. It can stop its own secretion by acting on the anti-pituitary, by acting on the hypothalamus. So, this is about the regulation of testosterone. Now, what will follicle stimulating hormone do? Now, follicle stimulating hormone acts on Sertoli cells. Follicle stimulating hormone acts on Sertoli cells. Now, Sertoli cells, they produce a protein called ABP, androgen binding protein. Sertoli cells, they produce a protein called ABP. What is ABP called as? Androgen binding protein. It is called androgen binding protein. Now, this androgen binding protein, what it will do? Androgen binding protein, it acts in the process of spermatogenesis and it helps in spermiogenesis process. Androgen binding protein along with the Sertoli cells, they control a process. What is it called? Spermiogenesis. Now, what is spermiogenesis? Spermiogenesis means conversion of spermatid into spermatozoa is called spermiogenesis. We know spermatid is like this. Then this gets converted into a head, neck and tail-like structure, no? So, this is called transformation, differentiation. This step, conversion of spermatid into spermatozoa, is called as spermiogenesis. And spermiogenesis is under the control of androgen binding protein, which is secreted by the Sertoli cells. And what is the other role of Sertoli cell? Sertoli cell secretes a protein called inhibin. What will Sertoli cell secrete? It secretes another protein called inhibin. Now, what will this inhibin do? What will this inhibin do? This inhibin can stop the production of follicle stimulating hormone. This inhibin, when the concentration is more, so we have to go like this. When the concentration of inhibin is more, so it can go act again at the same hypothalamus. It can go act on the anterior pituitary. It shows negative regulation here. it shows negative regulation here. So the inhibin produced by the Sertoli cell can stop the production of follicle stimulating hormone by asking the anterior pituitary to not to release FSH, or by asking hypothalamus to not to produce GnRH, right? So this is also called feedback inhibition only. This is also called feedback inhibition only. Understood? So this is a hormonal regulation of spermatogenesis process. So under hormonal regulation, we were talking about hypothalamus. Hypothalamus produces a releasing hormone called gonadotrophin releasing hormone that comes and acts on anterior pituitary. Anterior pituitary produces two gonadotrophins called luteinizing hormone and follicle stimulating hormone. Luteinizing hormone comes and acts on the testis, comes and acts on the Leydig cells. Then the Leydig cells will produce a male sex hormone called testosterone. The testosterone maintains the male reproductive system, and testosterone helps in spermatogenesis process also. If the levels of testosterone are high, then it causes feedback inhibition. Testosterone can stop its own control by acting on the anterior pituitary, acting on the hypothalamus. That is called feedback inhibition. Now coming to the follicle stimulating hormone. The follicle stimulating hormone acts on the Sertoli cells. Sertoli cells will produce a protein called androgen binding protein. Sertoli cells along with androgen binding protein helps in spermiogenesis. What is spermiogenesis? Transformation of the spermatid into spermatozoa, right? And they will be released out. This is one. The Sertoli cells also release a hormone called inhibin. This inhibin has again feedback inhibition. It can go act on the anterior pituitary. It can go act on the GnRH hypothalamus. Stops the release of follicle stimulating hormone. So this is about. With this, we finish the structure of male gamete sperm and the process of spermatogenesis. In the next lecture, we'll talk about the female reproductive system. So if you like the lecture, like, share and subscribe to my channel. Thank you.